McCleary, CEO and President of the CFIDS Association of America, and it's my privilege to kick off our 10th webinar of 2010 with such an esteemed panel on such a great topic of interest in the CFS community. Our topic today, an XMRV update, has been one of the most popular of our series. This is actually the second XMRV program, and we're lucky to have Dr. Lucinda Bateman back for a second time. And we're also grateful to have another expert, Dr. Vincent Racaniello of Columbia University, to share some background information about the virus itself and also his perspective on where CFS and XMRV research stands, followed by Cindy's presentation about how CFS, the landscape of CFS research has been forever changed by this discovery and the research that has followed. Before turning the program over to my colleague, Dr. Suzanne Vernon, our scientific director, I was just going to run through some housekeeping points to make sure that we're all oriented to this webinar software and that folks who have joined us today for the program understand how to submit questions and do other things. First of all, understand that each of the four of us is in a different location, so we have no way to send verbal cues or hand signals or anything to one another. So we're experiencing this almost the same way as everybody else is, sort of in our own little places without the benefit of body language. And it's a little bit of an odd experience. I think we're getting used to it at this point, but it's still a little strange to sit here and talk to yourself. We did receive many questions submitted with the registrations as people let us know of their interest in participating today. Those were shared with the speakers in advance, and we'll have time for Q&A after the slide presentations, and Suzanne Vernon will moderate that. We've kind of compiled the most frequently asked questions, and we'll probably lead off with those. And there was quite a lot of duplication in the questions that we received. Just a couple of points about the topics and the questions that you might submit during the program. The speakers are not able to address individuals' questions about personal symptoms or treatment or how one part of their medical history might connect with another part of the medical history on a personalized basis. This format just doesn't work well for that, and even though Cindy is an MD doctor, this just doesn't really work well in a webinar situation. Those are really clinical questions that need to be posed to your own physician. And also the subject of unpublished data cannot really be discussed. So Cindy will give us an update on the study she's involved with, but we're going to respect the investigators' right and privilege and need to publish that data before it is discussed. So also, just as a reminder, the recording of today's program will be posted online within a few days. We did have some technical issues with our last one that I think we have resolved, so hopefully we'll get that recording up along with the slides within a few days' time. And I think that about covers it. I'm going to turn this over now to Dr. Suzanne Vernon. Excuse me, she's our scientific director and has been with the association for 2.5 years. She's been a researcher in the field of CFS for, gosh, over 10 years now. I keep losing count, Suzanne, but you'll correct me. And she was at the Centers for Disease Control and Prevention before joining the association staff, and has recently participated in a meeting in Japan and shared some of those highlights with us last week. So look for her other webinars online. But she is a PhD who trained in virology and is going to help moderate today's discussion, including the Q&A session. So Suzanne, I'll let you take it away from here.
Okay, thank you, Kim. If you can bring up the next slide. I'd really like to thank and recognize, let's go to the next one, Kim. Recognize, oh, no, not that one either.
This one?
No. Yeah, that one. Thank and recognize the association's organizers of this webinar series. And I think it's been just really a neat, series of webinars on hot topics like the ones that we have today, and also the priority issues, the issues that are really important to all CFS stakeholders, patients, scientists, and the medical community. It's just been a great way to get CFS-specific information out there to people that either can't leave their homes or are too busy at the benchside. So it's just been a great venue for distributing information. This series today, or this webinar today, is, I think, a webinar that epitomizes our attempts or our efforts at translational research. This is going to illustrate to all of our listeners the challenges that we face in bringing benchside discoveries, those aha moments that happen at the benchside, to patient care, to impacting what happens at the bedside. This is not a direct from the bench to the bedside route. It's actually many, many steps that are required from that aha moment or that discovery to actually the treatment or the actual cure for, in our case, CFS. This is not, in addition to being a many-step process, it's also not necessarily a direct shot to that bench to bedside. And I think this is exemplified in the slide that you've already seen very quickly, hopefully, didn't get to digest it. So Kim, if you can go to that rather complicated picture slide, that one, which shows up in the left-hand corner a gene called PXE, and this comes from a paper written by Sharon Terry, who is the CEO and president of the Genetic Alliance. This is a paper published in Personalized Medicine. Her children actually have PXE, and so she's dedicated her life to trying to figure out diagnostics and cures for this rare genetic disorder. And she's done a lot of work with the NIH and a number of academic organizations to illustrate or to kind of catalog the numbers of steps that are required in order to get from a discovery, such as the PXE gene, to actual objective approaches of diagnostics, which is over there in the right-hand corner, and over in the bottom right corner is the therapeutic treatment or the interventions. So many different steps required. Again, not always a direct route from the discovery to the treatment or the diagnostics, and many different types of experts involved, as well as many different types of techniques involved. So it's a process, and I think what this indicates to all of us is that this process takes time, this process takes expertise, and this process takes, obviously, the support, both in the intellectual support and the financial support that we need in order to get from a discovery to the actual bedside implications. So I think this is what we're going to get a little bit of a sense of. Today we're pleased to have two speakers that represent both ends of this spectrum. Our first speaker is the benchside expert. Vincent Racaniello is a virologist and a professor in the Department of Microbiology and Immunology at Columbia University College of Physicians and Surgeons. His laboratory conducts research on poliovirus, so true benchside research. But I think importantly, what Vincent has done is dedicated a lot of his energy and efforts to educating people about viruses and virology in general. So not only students, but the public, and he uses this great medium with podcasts and is able to reach people all over the world. so beyond the classroom. And many of you, or some of you, may be familiar with this media. It's called TWIV, or This Week in Virology, and maybe Vincent will mention that later on. But you can learn a lot about virology and a lot about Vince by checking out his website and the TWIV podcast. Our second speaker is the bench-side expert. After graduating from Johns Hopkins School of Medicine, Dr. Cindy Bateman returned home to Utah, where she has practiced internal medicine since that time. I guess that was in the early 90s, right, Cindy?
Yes.
Yes. And in 2000, Cindy opened the Fatigue Consultation Clinic, where she and her staff care for more than 1,000 patients with chronic fatigue and pain. Cindy also established and is now the executive director of the nonprofit organization called OFFER. And in addition to caring for a bunch of patients and leading this nonprofit organization, she also spends a lot of time giving lectures and CME on CFS and fibromyalgia. So without further ado, I'd like to open up the mic to Vincent.
Thank you, Suzanne, and thank you, Suzanne and Kim, for having me. I would like to talk about the virus XMRV, xenotropic murine leukemia virus-related virus. And Kim asked me to specifically answer 3 questions. And the first is, what do we know with some certainty about XMRV, the agent? So let's talk about that first. First, how was the virus discovered? The genome of this virus, that is the genetic information, was first identified in surgically removed tumor-bearing prostates in 2006. And the virus itself was not actually recovered from those tumors, but rather the sequence of its genetic information. And that sequence was put together to make a DNA copy, which was then inserted into cells, and then the cells produced viruses. So these are cells grown in culture. And that was the first ability to look at this virus, which is shown here in this electron micrograph. So these are the virus particles of XMRV, and viruses, of course, are very small. This virus happens to be 100 nanometers in diameter, which is 0.0001 millimeters. Very, very small. And you need very special microscopes to see them. So that was when XMRV was first discovered. Here's a diagram of the structure of this virus. It is a typical, what we call enveloped virus. Around it is a membrane which is derived from the host cell in which the virus replicates. You can see it's called an envelope here. Within that envelope are viral proteins, and these are needed for the virus to attach to cells. And then within the center of this virus particle, this is a cross-section, if you will, is the core, which is basically a set of proteins that protects the genetic information of this virus. And the genetic information is labeled here as RNA genome. So this is a virus with a membrane and which has genetic information composed of RNA, ribonucleic acid. And viruses come in both flavors. They can have either RNA or DNA as their genetic material. This one happens to have RNA. Now this is a very unusual kind of virus. It is a retrovirus. It's a member of a family of viruses, all of which have a genetic information consisting of RNA, but which do a very unusual thing in their cycle of replicating. So on this schematic, we're showing a typical retrovirus particle on the lower left. It's in blue. It's attaching to cells and it's putting its genetic information in the cell. And the 2 black lines there are the 2 pieces of RNA that have all the information to make new virus particles. And what's unusual about the replication of these viruses is that they take that RNA and convert it to a DNA copy. And that's called reverse transcription, and that's why these viruses are called retroviruses. So you see the genetic information is going from black, which is RNA, to red, which is DNA. That's important because that DNA then goes into the nucleus of the cell and inserts into the host DNA, the chromosomal DNA of whatever cell the virus is replicating it. And in that state, it's called proviral DNA. And it's shown as a schematic in this picture as two red lines inserted into black lines, and the black lines represent the DNA of the host cell. Okay, so that is unusual for viruses in general. It's only members of this family of retroviruses that do that. And it has very important implications for all aspects of the viral life cycle. Now, in the next slide, we're showing how these viruses make more of themselves. So we've got a cell in which there is a proviral DNA already, and the cell unwittingly participates in this. The cell makes messenger RNAs, which direct the production of proteins that the virus needs to make new particles. So you can see here these mRNAs coming out of the nucleus. They're directing the synthesis of proteins that the virus uses to assemble new particles, which eventually are released from the cell as new viruses. Now, for some retroviruses, these new virus particles are very important in the spread of infection to other cells. So, for example, if this were a cell infected with human immunodeficiency virus, HIV, these new virus particles would go on and infect other cells and spread the infection. But for other viruses, there are very few virus particles, if any, made, and viruses spread from cell to cell as a proviral DNA, as a DNA copy that's transmitted to a new cell as the cell divides. So that's an important aspect of this life cycle to keep in mind. Another important aspect is that wherever this proviral DNA inserts into the host DNA can have pathogenic or disease-causing consequences for the host. So if this proviral DNA sits next to a gene which is normally not functioning in a particular tissue, it could turn on that gene and cause pathogenic consequences. So diseases caused by these viruses can be a consequence of the infection being spread by the virus, or they can be a consequence of where the proviral DNA inserts into the cell. Okay, this is not the only known human retrovirus. The first human retrovirus was human T-cell lymphotropic virus 1, HTLV1, which we'll talk about a bit in a moment. And of course, HIV 1 and 2 were subsequently discovered. And given the nature of HIV-1, which of course causes AIDS, antiretroviral drugs have been developed to inhibit that virus. And some of these are shown here. And in particular, some of those drugs have been shown to inhibit the replication of XMRV in cell culture. And these include AZT, raltegravir, ziduvudine, tenofovir, disoproxil fumarate. And these would have to be tested, of course, in clinical trials to see if they have any therapeutic value in XMRV infections. The second question is, what studies need to be done to understand the role of XMRV in causing human disease? Now, the connection of XMRV with CFS was first discovered in a paper published by Lombardi et al. Last year in 2009 called Detection of an Infectious Retrovirus XMRV in Blood Cells of Patients with Chronic Fatigue Syndrome. And a quote from that paper is, "We have discovered a highly significant association between the XMRV retrovirus and CFS." And they also said, is XMRV infection a causal factor in the pathogenesis of CFS or a passenger virus in the immunosuppressed CFS population? And in a review paper that accompanied that publication, Coffin and Stoy wrote, whether the virus plays a causative role in either CFS or prostate cancer is unknown. So what do we need to do to answer these questions? A very important series of studies that have to be done are case control studies. These are studies where you have cases of individuals with a known disease. You match them with controls who are similar in most aspects except for having the disease, and then you ask what's different between the two populations. These have
Vincent,
to be multi-city. Yes.
Can I stop you for a second? Is there any way you can turn your volume up?
I can, yes, of course.
Several people have written in and said they're having a
low?
hard time.
Okay.
That's better.
Is that any better? Okay.
I think it is.
Okay. Let me know it right away if it's not. I can
Okay. Thank you.
pick it up a little more.
Sorry
Sure,
to
I'm sorry.
interrupt.
No, it's fine. So these case control studies need to be multi-city and multi-continent. They have to use well-defined patient populations, and the virus must be consistently identified in patients with the disease, either prostate cancer or CFS. And besides making the association between the virus and the disease, we'll learn other things from these studies. We'll learn about how the infection is transmitted by taking very careful case histories. We can determine the seroprevalence of the virus in healthy persons, what fraction of the population is infected, and then, of course, of those, which develop disease. Is everyone who is infected develop disease, or just a small fraction? And with viruses, it can go either way. Some never develop disease after infection with certain viruses, and for some, the infection is 100% causing disease. We also need to develop assays to detect infection, and we need multiple assays. We need assays perhaps for virus, for proviral DNA, that's the DNA integrated into the host cell. We need assays for viral proteins, and we need assays for antibodies to the virus. So why do we need more than one? Well, a good example is AIDS. If you have an AIDS test to see if you're infected, if that test is positive, a second different test is immediately done to make sure the first wasn't a false positive. So we have to have multiple ways to look for the virus. Having such an assay will give us other important information. It lets us determine where the virus is replicating, what kinds of cells and what kinds of tissues the virus is replicating in. And that's important because we need to know if there is a cell that is infected by the virus that would explain the kind of disease that we see. For example, in AIDS, the main cell that is infected is the CD4 positive T lymphocyte, a white blood cell. The destruction of those cells leads to the immunocompromised state and opportunistic infections. So we know the virus replicates in those cells, it kills them, and that causes the disease. And also, we would learn from these assay results whether there is a consistent integration site of the proviral DNA that would explain the pathogenesis. So if we found that the provirus is integrated in similar sites in different individuals, it would suggest that it's near a gene whose activation is involved in the disease. So here is that illustrated one more time. The proviral DNA is inserted into the host chromosomal DNA, and where it inserts might be having important consequences for the disease, and that's one of the things we need to learn from having better assays for infection. Now, so far there have been a number of studies examining the presence of XMRV in either prostate cancer, chronic fatigue syndrome, and amyotrophic lateral sclerosis. And the results have been mixed. Some of the studies detecting the virus in prostate cancer, some not. And in terms of CFS, the initial Lombardi study detecting the virus in 67% of CFS patients, but 4 subsequent studies not detecting it. And the fourth study, the fourth negative study, recently published from the CDC by Switzer et al. So why do we have such inconsistent detection of the virus? There are a number of possibilities. The selection of the patient populations may differ. There may be geographic differences in the distribution of the virus. This is well known for other viruses and is completely possible. And hand in hand with that, it may be that there are sequence differences among the viruses in different locations, and that may preclude detection. But perhaps the most important issue is that there's no standardized sensitive method of detecting the virus, and there are no positive control specimens that have been used, and this needs to be done. So let's look at this in a little more detail. In the Lombardi paper in 2009, they used polymerase chain reaction to detect proviral DNA in peripheral blood mononucleocells, lymphocytes, and macrophages. Subsequent studies, the negative studies that I showed on the slide, have used a similar approach. Now, it's been suggested that in vitro culture of infected PBMCs is important for amplification and detection of the virus, but this has not been published, and therefore other studies are employing the methods described in the Lombardi paper. So this method needs to be published because a scientific paper should be sufficiently detailed that anyone can reproduce the findings, and if there's some differences, those need to be added to the literature. We can learn a lot from other viruses, though, when we think about XMRV, and one is HTLV1. This was the first human retrovirus to be discovered. It's the causative agent of adult T-cell leukemia, lymphoma, and myelopathy, known as tropical spastic paraparesis. The virus infects CD4 positive T lymphocytes, but it's transmitted from cell to cell, not by free virions, but as the proviral DNA. And when you're infected with the virus, either through the blood supply or through sex or intravenous drug use, the virus comes in a cell as a provirus, not as a free virus. And it multiplies, the virus multiplies not as a free virus particle, but as the cell divides. So this is very important because it's likely that the pathogenic effects of this virus, the leukemia, the myelopathy, are due to where the provirus has integrated into the cell. And it's spread again within you by the cells multiplying, not by free virus particles. HTLV1 is diagnosed by detecting antibodies in serum by ELISA, and any positives are confirmed by Western blots. So again, 2 assays are used. We don't use virus isolation for diagnosis. There's very little virus present. A PCR assay has been developed which can be used by clinicians. It's very sensitive. It can detect 10 molecules of viral DNA per 1 milligram of human DNA. That's a lot of DNA. That equates to 1 infected cell per 100,000 PBMC. So the point here is that if you need to have a very sensitive PCR assay, it can be done. The way the infection is treated is that there are no antivirals to treat it. There's no proven antiviral therapy. We really don't understand the role of the virus itself in human disease pathogenesis. So we don't know if we eliminated the virus, if that would have any impact on disease. And as I said, it could be that the insertion of the provirus is the main cause of the disease, so eliminating any virus present would have no impact on clinical symptoms. HIV/AIDS is another good example to learn from. It's very different. Infected individuals have millions of virus particles per milliliter of blood. This makes it very easy to detect infection by looking for antibodies or viral proteins or even viral RNA. And we can treat these infections because antiretrovirals work by limiting the virus production. The viruses produced infect new CD4 positive cells, and that's the cause of the disease. So if we limit the virus production, we can control the disease. We also need an animal model for virus infection, for XMRV specifically. These have been essential for understanding the pathogenesis of many viral infections. They may provide information on what cells and tissues support viral infection. But remember, they may or may not develop symptoms that mimic human disease. They are useful for testing vaccines and antiviral drugs. But they're called models because not everything we learn from them will be the same as what happens in humans. Now, nothing has yet been published on animal models of XMRV infection, but at meetings there have been some studies in rhesus macaques that have been reported where these animals are inoculated intravenously with virus and sacrificed at different times after infection. The key points are that the virus seems widely disseminated in many tissues, and there's very little virus in the plasma. It seems to infect lymphoid cells and the prostate, seminal vesicles, epididymis, and testicles, and also cells in the cervix and the vagina. But again, we don't know if this will be the same in humans. It gives us guidelines which we can then validate in humans. And finally, the last question, when viruses are first associated with human disease, how many studies does it take to prove or disprove association? And I added the word causation here because we'd also like to know if the virus causes the disease. Again, I think we can learn a lot from history, and AIDS is a good virus to learn from. The virus, the history of AIDS begins in 1980. In December of that year, there are unusual clusters of opportunistic infections that were in populations where it hadn't been seen before. That led to multi-city case control studies in the U.S. in 1982, run by the CDC, and these provided evidence that there was an infectious agent involved and that it was spread by blood or sex. In 1983, by the end of 1983, 1,000 AIDS cases had been documented, so that's 3 years after the first initial signs. In July 1984, the virus was isolated, and in February 1985, the first antibody tests, which were ELISAs and Westerns, were developed. The first drug approved for treatment was AZT in 1987, and in 1995, highly active antiretroviral therapy using a combination of 3 drugs was begun. So 15 years from the first suspicion of AIDS to having a very good way to control the disease. So it takes time. HIV-1 and 2 is a virus that jumped from non-human primates to humans, very much like XMRV has gone from mice most likely to humans. But the AIDS story was not without controversy, and even today, with tens of millions of AIDS cases globally, some individuals still claim that the virus does not cause the disease. HTLV-1 is another virus that we can learn from. In the late 1970s, it was first isolated from the blood of a patient with T-cell lymphoma. Its causative role in the disease was established by independent case control studies in the U.S. and Japan, one in 1977 and 2 each in 1981 and 1982, so over the course of about 5 years. The virus is readily found in people with the disease. In 1986, it was associated with a second syndrome, myelopathy. And as a consequence of this virus and the disease it's caused, donor blood in the U.S. and Japan is screened for the presence of the virus. And like HIV-1, it jumped from non-human primates to humans. And here is a point I want to emphasize. In 1985, a separate type of HTLV was identified, HTLV-2, which is in people but not yet associated with human disease. So an example of a virus that can be present in humans but not cause a disease. And that leads me to my last few slides, which I've entitled Koch's Postulates for the 21st Century. Robert Koch was a German microbiologist in the 1800s who figured out how to determine if a bacteria caused disease. And today we have very different detection methods for pathogens, so we've had to change the way that we think about this. And so these are the 21st century version of his postulates. The first is that a nucleic acid sequence belonging to a putative pathogen should be present in most cases of an infectious disease. The nucleic acids, in this case, say XMRV, should be found preferentially in organs known to be diseased and not in organs that lack pathology. Fewer or no copies of this nucleic acid, again, think XMRV, should occur in hosts or tissues without disease. When the disease resolves, which may happen in some cases, the copy number of the nucleic acid should decrease or become undetectable. With clinical relapse, when the disease returns, the opposite should occur. When you can detect the sequence of the virus before disease, or when the copy number of the sequence, how many viruses are present, correlates with severity of the disease or even pathology, this gives more evidence for a causal relationship of the virus or the microbe with the disease. It would be good to demonstrate the sequence in areas of tissue pathology or areas where
Vincent?
Yes.
There's some kind of weird vibration feedback coming through suddenly.
I don't hear anything. Sorry, it just started. Yeah,
it started maybe at the end of the last slide, just a minute or two ago. Sounds like you're underwater.
Okay. Let's see if I can fool with the volume here. Yeah, I got. That's the best volume I can get here. Well, I'm almost finished. So we should demonstrate that the sequence is in the area where we think there's pathology, where the virus should be. And finally, we should be able to reproduce these sequence-based forms of evidence for causation. So those are 6 points that we need to fulfill today. So that is what we need to do to prove that XMRV is a causative agent for CFS. So I have taken, sorry. I'd like to end it just by sending you to my blog, Virology Blog, where I have a goal of teaching everyone about viruses. You can find it at virology.ws. I try and explain viruses to the public in a way that's understandable by anyone, irrespective of their science background. And we have written posts as well as podcasts this week in virology, which I think you'll find interesting. We cover all viruses, and of course we also cover XMRV and its role in human disease. Okay, thank you very much.
Great, and there were quite a few questions that came in during your presentation, which I will try to send over to Suzanne. And if you could change your controls over to Cindy.
Mm-hmm.
I can do that.
Hey, Vincent, could you say something again? It seems like we might have lost your echo.
Hi, I'm Vincent Racaniello. Am I still echoing?
No, it's perfect now.
Now that I'm done, it's perfect, right?
It was only the last couple of slides. You sounded somewhat robotic.
Oh,
I think there was an echo.
Yeah. All right, Cindy, are you ready to take over the controls?
I guess so. I just wanted to say that I lost internet contact twice during his talk, so I've ordered everyone in my office to stay off the internet, and I'm not sure why that happened, so hopefully it won't happen again. Well, it's a pleasure to be here, and especially to share the time with Dr. Racaniello. As everyone knows, I'm very involved in the clinical aspects of CFS, and everything that goes on in the field is important to me. So it's a pleasure to be here. And you know, when I scheduled or agreed to do this webinar many months ago, I'd hoped to discuss the research going on in Utah, but I have to admit that it's not done and I can't talk about it. But I am certain that you will hear about it in the future. The research team involved is Dr. Ila Signh and her XMRV research team. Also Dr. Allen and Kathy Light, who've worked on some other CFS projects, and then well-defined patients from my clinic. And I put well-defined in parentheses there because I'm going to talk today about how difficult it is to decide when you have a well-defined patient. So hopefully that will shed some light on some of our difficulties. You know, it's important to remember that in science, silence is a very good thing. When people have disappeared into their labs to work, that means that some good science is going on, and we've had our heads down since March. So I'm certain that our study will be, I'm confident it will provide some helpful information for the field when it appears. So as I was considering what I was going to speak about today, I sat back for actually several weeks just thinking about what I've learned in this time period since the first study showing XMRV was related to CFS or could be related to CFS. And I decided that actually many good things have come from what's been somewhat painful controversy surrounding XMRV and CFS. And I'd like to share with you 7 different concepts or things that have come from the controversies surrounding CFS. The first is, and we know these negative studies have been painful for those of us who would like to see this come to be. But what I've learned is that a critical analysis of every study, even the negative studies, can improve the quality of our testing methods and help us choose the best CFS population to study. And you may have read a critical review by Dr. Vernon regarding the negative CDC study that was just published. And just make a few comments that this study used a patchwork of patients from several different studies and probably used at least 2 different CFS case definitions. And there was a relatively small number of patients included, and probably the broadest CFS case definitions that exist were used to define the patients. So it's like looking for a small fish in a very big pond. In addition, it's possible each study used different blood collection tubes and they didn't match the tubes used in the science paper, but either way, each paper that's published helps us re-examine the way we're doing science, the way we're looking at patients. I've had the opportunity in the last 2 months to attend XMRV Journal Club at the University of Utah. And this is a process that occurs among researchers in an academic setting where they gather together, read the most important or pivotal papers or most recent papers, and try to analyze the content of the paper so they can go on and do even better science. And the most senior scientists in the room, when we were finished, said, with each paper the bar will be set higher. And this was a challenge to her team, and this is my team also, that when we do research, we're going to have to, every time there's a paper subject to review or when it's published, we're going to have to do a better job each time. And that's what comes out of papers. So we shouldn't be discouraged when papers don't show what we want. We should take the information from the paper that's going to teach us the most, and progress will march forward. All right, the second good thing, and this is a complex one, but it's a big subject for me, there is finally more serious scrutiny of the way we define CFS. Now, we all know people in the field of CFS and patients have been arguing for a long time about how to define CFS. And to be honest, no one cared. No one outside of the field really cared how we define CFS because nobody cared about CFS. But now that there's an important reason to know how to choose patients, we realize that our case definitions are a problem. But we should understand rather than argue about the various subjective ways of diagnosing CFS and ME, FM, MCS, all of these related conditions that we define subjectively. If we could analyze what we learn from the different ways of defining patients, just like those scientists in Journal Club analyze the papers, we'll be able to learn a lot more about this population. And I hope I don't get anybody too upset when I say this, but there is no true or pure CFS. I have heard scientists and people use that term, pure CFS. What we have are our various educated but biased opinions about CFS, and too many ways to subjectively define this large group or groups of sick people, and several yet to be objectively defined subsets. So we need to be more precise and more objective in how we define our subsets. So these case definitions were good to start with, but as we need more specific information and as we're looking for specific objective markers, we'll understand the shortcomings of our definitions. And we just need to understand them and move forward. Now, you wouldn't argue that this is a pretty big snake. This is my living room, by the way. You can see it, you can feel it and hold it. You can feed it, weigh it, you can look it in the eye and hope it doesn't want to give you a bite or a hug. That is easy to define. But let me just present some challenges to you about what it means to define CFS. Which is CFS? The Fukuda defines CFS. This is a research definition that includes both gradual and sudden onset. It excludes people with conditions that might contribute to the fatigue. It doesn't require post-exertional malaise or relapse. We know that it defines at least 1 million people in this country, and it's been a very important definition. It has its weaknesses. We know that genomic studies show that gradual versus sudden are two distinct groups of gene expression. And I can tell you that many patients with fibromyalgia, if they have significant fatigue, they meet the Fukuda's case definition. So we have some problems, and it's very broad. Then the empiric case definition of CFS, we know has been met with plenty of controversy. It broadens the group defined by Fukuda by at least 4 times, estimating that 4 million people in the country would meet this kind of a definition. And as Dr. Lenny Jason has asserted, it probably not only includes people with other conditions like fibromyalgia, but many patients with depression. I think that my clinic, as a clinic for evaluating chronic fatigue, probably reflects the empiric case definition, a broad group of people with unexplained chronic fatigue. We have the Canadian definition, which is a good description of symptoms. It's inclusive. It's great diagnostic and treatment guidelines, but to be honest, only CFS specialists have heard of it. It's hard to access. It's not fully recognized or utilized beyond the clinical community of CFS. Doctors in the community who don't specialize in CFS have no idea what it is. So there's other problems, other confounders. In the Georgia study that projected 4 million people in the United States with CFS, they also identified another 4 million people who seem to have CFS or have CFS-like illness, but have diagnoses that haven't been identified or treated. These are people who need a good doctor, and a doctor who can evaluate them over time. I know from my experience in my clinic that many times my patients end up having atypical presentations of sometimes difficult to diagnose conditions, or conditions that may not meet established diagnostic criteria or can't be diagnosed yet. For example, patients who have MS, they may have symptoms for a decade before a diagnosis can be made. And there are atypical or mild or different presentations of autoimmune diseases, and there are always rare genetic or neurologic conditions that may take years to get a diagnosis or never if they don't meet the right clinician. I'll give you an example from my own clinic. I had a woman in her late 60s that I evaluated recently. She had horrible fatigue and exercise intolerance. It started during a time when her life became very stressful. There was a flu syndrome involved. I actually included her in our study, even though I hadn't known her very long. But after a few months of detective work, just careful history and working with her to try to help her feel better and doing diagnostic studies, we discovered that her Cymbalta was making her tired. There are about 10 or 15% of people who become very tired and sleepy on Cymbalta. And I had her do an exercise test because of her age, and it turned out she had coronary artery disease. And because she had an atypical presentation, she didn't get chest pain, but she felt very ill when she tried to exercise. So I guess the point is that hidden among our group of patients with who meet the criteria, there are people who have other conditions, even in inexperienced hands. There are also patients who have what I call complex chronic fatigue. That means they have many different conditions that contribute to fatigue. Sometimes they have CFS, sometimes they don't. I had a patient who I'll call a skinny CFS teenager that I took care of when she was 18. Then I didn't see her for 10 or 15 years. And when that time had passed, she came back to my clinic as a woman in her 30s. She was unrecognizable. She had gained 150 pounds. She was disabled. She had heavy medication dependence, chronic infections, poorly controlled diabetes, severe sleep apnea, and of course she was depressed and hopeless. She can barely leave her house. What does this woman have? Should she be included in studies of CFS? I know she has CFS as an underlier, but I also have patients who don't have the history of CFS who present with complex medical problems. Should they be excluded? Should they be included? Other confounders we might consider. When sometimes as clinicians we use a subset definition when we're defining patients in a non-standardized way. That means we just do it in our head when we're picking patients for a study. For example, we might apply a Holmes-like, that's the 1988 case definition, where we might not call a patient CFS unless they have sudden onset or have very strong flu-like symptoms. Some researchers might only include those with decreased natural killer cell function or abnormal RNA cell. This is my own bias. Consider those only whose post-exertional relapse is severe or of a certain nature, or who relapse more easily. I just use post-exertional relapse as a diagnostic tool for CFS, but it's not required for the ways we define CFS. It is in the Canadian case definition. How about only counting those who are very impaired, and don't count those who meet the criteria but can function at a higher level? I've heard clinicians say that if you don't have cognitive dysfunction, you don't have CFS. Well, that's definitely a process of subsetting that isn't the way we define the illness. I'm not saying they're not valid. I'm just saying we have no consistency in the way we diagnose, the way we choose patients sometimes. And the last example I have is including only patients with evidence of elevated antibody titers, like HHV6 antibodies or EBV antibodies, or a certain cutoff for how high their antibody titers should be. Should we use those as a way of subsetting patients? Neither is right or wrong. It's just not the way we have defined the condition with our more official case definitions. Other confounders. CFS impaired people go on to develop comorbid conditions, sort of like my example of the young teenager. Most people with CFS as they age get sleep disorders, They may get diabetes or have depression, age-related problems, a coronary disease like my patient. Should we include them or exclude them when we do studies of CFS? What about comorbid conditions that arise or may arise directly from the disease state of CFS? What about those lymphomas in Nevada? Those patients that may have some kind of a chronic viral infection and go on to develop malignancies. Should we exclude them or include them when we're studying CFS? We know that there's relative deficiencies of hormones in patients, HPA axis dysfunction. Should we exclude them because they have another disease or include them and assume this is something we've learned about CFS? Jerky or restless legs. Almost all my patients develop mild myoclonus. My teenagers get mild myoclonus, and many people have problems at night. This could be considered an exclusionary diagnosis, a different diagnosis that impacts sleep and causes fatigue. Should they be included or excluded? What about different types or causes of CFS related to age? Or are these stages of CFS, or are they true subsets? Let me give you some examples. I see CFS in adolescents after Epstein-Barr infection. I call it post-DBV CFS. They have POTS, postural orthostatic tachycardia syndrome, and orthostatic intolerance, headaches, and disordered sleep. They're a very distinct group. They tend to get better more readily. They don't have as much cognitive dysfunction, but they certainly meet the CFS criteria. Are they different than a middle-aged active person who is devastated by a sudden onset flu syndrome and never recovers? They both meet the criteria. They may be two completely different kind of scenarios. They both result in something like CFS. And then still another example, someone who has stepwise onset. They're kind of fragile, and every time something happens to them, they get worse. They get an infection, they get better, but it takes them a long time to recover. They never get all the way back to the way they were before. And each thing adds to their pathology until they're very ill and in a place where they're very debilitated. I mean, these are just 3 examples of different subsets I see clinically. So these our case definitions just don't help us sort out these different subsets. They're a good start, but I find them inadequate to do research, too broad, too subjective, and they define heterogeneous group. So hopefully, good biomarkers will gradually change or end the debate about how to subjectively define CFS. I mean, maybe we can move beyond these case definitions if we have good biomarkers. And this is one reason we must continue to study all biomarkers, not just focus on proving or disproving XMRV. This may be our biggest challenge, defining a case of CFS, but it will also be our biggest triumph because it will help us move to the next level of understanding all the people who are ill with CFS. Okay, so the third good thing, the second was about recognizing that our case definitions are inadequate. The third good thing is the controversies are enticing more scientists to engage in XMRV and CFS research, and it's pushing those in the field to do higher quality science. Man, each study is critically scrutinized, and it's painful, but this will be good in the long run. But XMRV research has been infused with life. These scientists who were kind of in the background studying XMRV have been thrust into the foreground, and it will increase our knowledge and encourage funding. Questions raised about assays, contamination, or other methods will push the bar higher in terms of quality. And scientists who previously knew little or nothing about CFS are now asking good questions about CFS and learning more about CFS, regardless of the outcome. I mean, think about it. The national and international blood bank scientists, a broader group of virologists, the Department of Health and Human Services agencies besides the CDC. And I just wanted to tell you about one volunteer that I encountered. This was a woman, a microbiologist who worked in the lab and decided to help us with our study. This when we started our study, she did she did the blood draw of 100 patients and helped the patients complete all their paperwork. Actually, many more, because she also helped evaluate the control patients. So she spent a great deal of time getting to know these people and helping them engage in the study. Afterwards, she was so moved by this experience that she set up time to shadow the providers in my clinic. She spent many days in our clinic just learning from people with CFS about their disease. And one afternoon I walked into the office with my physician's assistant, and this microbiologist was sitting there. She turned to me and said, "We have to help these people." And I said, Yeah, that's what we're doing. But it was a pleasure to see the light bulb just go on in her eyes. And as soon as she was exposed, we had someone who will forever be committed to helping and pursuing good science with CFS. So this number 4 is related, and that is the interest in studying CFS is growing in major U.S. academic centers. Now, academic centers, that's important. There's not only an interest, but a renewed sense of urgency. And previously, it was only simmering, dormant, or absent, or pushed back into the corner. I mean, people who study CFS had to be in private practice or stuck in little underfunded academic niches where nobody paid much attention to them. But now, Stanford, Harvard, Cornell, I should say add Columbia, the Cleveland Clinic, Emory, Johns Hopkins, other universities across the country have been starting to do research and learn about CFS and try to jump into the fray. Now think about this. Universities are where new clinicians and scientists learn about medicine and diseases. That's how we impact and change attitudes. Medical students, residents will be exposed to CFS, young researchers, and they will take that knowledge with them in their career. And we will exponentially expand what we need to do in the future in terms of the science of CFS. Number 5, the 5th good thing is that pharmaceutical companies are interested, engaged, and poised to act as the science unfolds. Pharma will rapidly push the science forward in certain ways and will bring vast financial resources to the effort. Pharmaceutical companies not only develop and test drugs, they teach prescribers and consumers about the disease through mass marketing. That's how they sell their drug. So think about the difference in the last 3 years since 3 drugs have been FDA approved for fibromyalgia. Fibromyalgia has been transformed from an embarrassing word to the most common widespread pain condition. There are articles in every journal and ads in every journal. Every physician has been exposed to the term fibromyalgia simply because drugs have been approved by the FDA. So the interest of pharma may make a big difference. Remember, there are potential treatments for XMRV infection, but questions remain regarding the role of XMRV in any disease, not to mention which treatments will be effective and safe. But we may get a jump start from the interest of pharma. The 6th good thing is that hope that progress is on the horizon has inspired donations to research from the private sector. People with financial resources who have CFS or who are the loved ones of people with CFS are writing checks like never before to fund CFS research. This is very important, and it may provide just the leverage needed to increase the seriously inadequate funding from federal sources. It is, sorry about that, it's a snowball effect, understandably. So the better the science, the more the funding, the better the science. So I would encourage interested parties in the private sector, if you want to donate, you should donate broadly to CFS research and biomarkers, and it will help push us forward. The last great thing is that XMRV has stimulated great interest from the media. Now, this is crazy making for the researchers. I can't even imagine what it's been like to be Dr. Harvey Alter the last month. It is difficult. It makes it difficult. It creates an uncomfortable situation and sometimes can sabotage studies. But on the other hand, the media interest keeps the CFS cause alive and will generally educate all about the impact of CFS on individuals and on public health. Now, I want to just leave you with an image. I should have put one more slide in, but I didn't. But imagine in your mind a turtle or a tortoise or a sea tortoise. This is the symbol we use in my office to remind patients how to live with CFS. Be the tortoise, not the hare. But, you know, this also applies to Cindy?
I think we lost Cindy.
We lost Cindy.
Not good.
Are you there, Cindy? Kim, can you see if that was her last slide?
It was her last.
Moves forward.
Oh, there she is. We lost you for a second, Cindy.
Cindy. Lost her again. Maybe we should go into some questions, Kim?
That sounds good. And if she comes back, she can tell us about the sea turtle.
Okay, or the tortoise, not being the hare. Vincent, are you there?
Yes, I'm here.
Okay, super. We have a number of really interesting questions and relevant questions. So I've kind of divvied them up between ones more relevant to you or Cindy or to the both of you or to us all. So let's start with this one, which I think is a really good one. Would XMRV be detected by the ViroChip? Are you familiar with the ViroChip, first of all?
Yes, yes I am. Oh yes, it would be. In fact, that's how XMRV was first detected in the prostate tumors in 2006 by using a ViroChip. So those sequences, they reacted with sequences from related viruses on the chip, and so the answer is yes, it would be detected in that way.
With that original detection, that was done with prostate tissue then, right?
That's correct, yeah.
Okay. Okay, I wonder if anyone's tried it with peripheral blood cells on the ViroChip. Do you know that?
I don't know that, but if you now know the virus, the specific virus that you're looking for, then it's less, it's less compelling to use a ViroChip. You can use other methods.
Very true.
So I'm not sure that people are doing that.
This is another good question. How do scientists know the difference between contamination in the lab and true positive results when looking for viruses? And
yeah, that's a good question.
You want to give an example of how, maybe an historical example about how contamination, a result was reported that actually was the result of contamination?
Well, I don't, none come to mind right now, but with XMRV we can use some of the results to answer that question. And one of the interesting observations is that every XMRV isolate from each individual patient is slightly different, which would tell you that this is not simply a contaminant that was introduced into the samples, because in that case every isolate would be the same. So that's a very good way to determine whether you have contamination or not, is simply to determine a good part of the sequence of the virus and see if it differs among different people. So in the case of XMRV, we believe that XMRV originated from a murine leukemia virus. It's quite different from any known murine leukemia virus, so it's not a contaminant of one of those. And each isolate seems to be different. So that's really the best way to address that.
Good. There is a question about how many XMRV studies are being done now, and I guess I would say that there's quite a few. And how many of those are actually using the WPIs or the methods that were described in the science paper? I would think that that would actually vary quite a bit as far as the actual methods being used. Do you have any more information about that? Methodology, WPI's methods, numbers of studies being done, or are a number of groups trying to optimize their own methods?
I would think that it's a combination, as you say. So some will take the methods as described in the science paper and try to reproduce them, and others will use their own approach, which is the way science works, right? You might find a better way.
Right. You touched on this one a little bit. Do we know that blood is the right thing to be testing for XMRV? And what about other tissues?
Exactly.
What should we be testing?
Well, that's why we need some extensive case control studies to find out what would be the best tissue. The blood would be very convenient, obviously, but it might be that it is somewhere else and more easy to detect. So we don't know the answer. It's a very good question.
Do we know if blood samples have been tested in patients that are XMRV positive with prostate cancer or prostate cancer XMRV positive patients? Do we know that?
I don't know. I don't believe it's been done, but I don't know whether the blood has been looked at. Yeah.
Okay. And you also mentioned the need for an animal model, and you described some unpublished results about some non-human primate research that's going on. What other animal models, in your opinion, would be most useful for the study of XMRV?
Well, a non-human primate is very difficult, as you know. They're very costly and difficult to maintain. Historically, in virus research, mouse models have been very useful. Now, this virus, XMRV, does not infect mice. They are resistant to infection, but you can easily make them susceptible to infection again genetically. You can make mice with a specific gene encoding the receptor for the virus, and that would make the mice susceptible. So I would bet that in some lab somewhere someone is doing that, and that perhaps that would give us a very nice, convenient small animal model to use for studying this infection.
Mm. This is somewhat of a philosophical question. What happens if XMRV research doesn't pan out for CFS? Do you think this is the end of the enthusiasm for investigating CFS?
I don't think so. This is a new human retrovirus. There aren't very many of them. They interact differently with humans, with animals, than other viruses do. So I would expect that research will continue to try and understand what this virus does. So irrespective of what happens with CFS, I would guess that the virology community will continue to work on the virus.
Yeah, I would agree with that. What do you think it means or signals, if you want to venture a guess, look into your crystal ball, what do you think this would signal for CFS research if XMRV is not replicated in any other study?
Well, you know, prediction is very difficult, especially of the future.
So you're not investing in the stock market, I take it, huh?
I think, as Cindy said, this has invigorated and revitalized the entire community around CFS research, and there are people thinking about it that didn't before. So that is a good thing, and I think that in the long run, no matter what happens with XMRV, that base will be there, and that will probably be very useful in understanding what's going on.
I absolutely agree with that, and I think it provides us an unprecedented opportunity to really take a number of the biomarkers that have been described in CFS and move forward on those fronts too. I think the profile, the hype, the recognition that XMRV has given to CFS really provides an opportunity for us to forge ahead with the many other areas that are important and have been demonstrated in CFS. So yeah, it is truly an exciting time. One more question that I had down for both of you, but I'd like you to also maybe give it consideration. Are cancers like lymphoma and leukemia possibly linked to XMRV infection because it is a retrovirus? And as you showed in your lessons learned for HTLV1 and HIV, it in fact could be. So what do you think about the possible link between XMRV and some blood cancers?
Well, in theory, sure, it could be linked because it's a retrovirus, very much like HTLV1. And so it could be responsible for a certain fraction of leukemias or lymphomas. That would be something to look at. You could do case control studies to specifically address that question. Now that we have the virus, it would be very easy to do.
Agreed. The rest of the questions are very clinical questions that maybe we can have Cindy addressed, and we'll perhaps post them on some of our frequently asked questions on our website.
She's back, I hope. Cindy, are you back?
She's muted.
She's not showing up as muted on my screen.
She typed the message, "Please unmute me."
We have her. Oh, gee.
Hey, Vincent, since you're very media savvy, at least when it comes to TWIV, I think this is also an interesting question. What makes XMRV more appealing or so appealing to the media other than some of the other biomarkers that have been published on and described as related or associated with CFS?
I think it's quite clear that viruses capture everyone's attention, right? The media loves viruses, especially the ones that make you very sick, like Ebola and Marburg viruses. Whenever there's a new virus, the media loves it. So this is a very concrete entity. It's a virus that might do this, and so very easy to wrap the head around, as opposed to, NK cells, which the media probably doesn't quite understand how that would play into a disease like this. So I think it's all in the microbe. The virus is appealing, and that's why it's taken off.
And so we have in the past had, retrovirus sightings in CFS in the early 90s, and enteroviruses have been detected in CFS patients. EBV is known to trigger CFS and cause post-infectious fatigue syndrome. Q fever, the Coxiella agent, is known to trigger a post-infectious fatigue state. What is it about the XMRV?
Well, perhaps being a retrovirus helps because everyone knows what HIV-1 has done.
Yeah.
I think that has had more press probably than any other virus, right? And probably the general public may know more about that virus than any other. So XMRV being in the same family, a journalist would look at it, oh, a retrovirus like HIV, it must be involved in a disease, right? So I think that's why. But I also think that the discovery has happened now in an era of massive communication, much more than has ever happened before. And information spreads incredibly quickly. People have the ability to make their own information, with websites and blogs. So this is totally unprecedented in history, and I think that XMRV has fallen right into the lap of that ability, and that's why everyone is taken with it.
And yeah, thank you for that. I think in many ways that's much to our good fortune for the opportunities for research yet to come in CFS. I'd like to really thank Vincent and Cindy for joining us today and sharing information about XMRV, lessons that we can learn from other chronic viral infections and chronic viral diseases, as well as the many challenges that we face in CFS research and in CFS medicine because of the natural history of CFS and the generation of many comorbid conditions that people with CFS end up encountering, as well as the diagnostic and therapeutic challenges that we face in CFS because of the lack of standard case definitions. This really does highlight some of the areas that I think just are screaming for focus from the medical and research communities.
Unmuted.
Thank you once again, everyone, for joining us. Cindy, are you back?
Cindy somehow is muted, and we can't figure it out on this end. So I deeply apologize because we had a lot of great questions that were clinical, particularly about testing and treatment, that I know she's probably screaming on her end.
Yeah.
She's ready to answer. Gosh, this is really frustrating, and I'm sure it's frustrating for the hundreds of people that are listening eagerly to hear from her about these issues, because they were some of the most asked questions beforehand and also during. She just typed a message that says, "My final word is slow and steady wins the race." And she's here, but we can't hear her, and I apologize. I can't figure out why that is happening. Vincent, any tricks I should be looking at? Since Citrix was a TWIB sponsor there for a while.
Well, a few minutes ago I was muted too, and I just clicked the audio input icon a few times, and that unmuted it. So perhaps you could try that, Cindy.
Don't be afraid to click, Cindy.
Yeah, just click away. You've
tried it many times, she says.
Yeah.
So that's not working.
All right. Well, some of the questions that we will have Cindy think about, and perhaps we can address them in another webinar or on our frequently asked questions, are about treatment. Should XMRV positive patients think about antiretroviral therapy? Should I tell my doctor about XMRV, and what do I do if I do test positive? And if I am positive, should I be concerned about various modes of transmission, such as sexual transmission? So we will try to work with Cindy, get some of these questions answered, and we'll perhaps post them on our website. How's that sound, Kim?
That sounds like a plan.
Okay. Thanks again, everybody. Thanks, Vincent. Thanks, Cindy. Thanks, Kim. And thanks to all our listeners out there. Let
me just make a pitch for our upcoming webinars, because a couple of them are germane to this topic as well. On August 12th, we have Louis Katz, who is an expert in transfusion medicine and the blood supply, coming on to talk about blood safety and XMRV. Then we're going to switch gears for a couple of programs, and Max Sasser, who is a disability attorney, will talk to us about Social Security disability in particular, and kind of the nuts and bolts of applying for Social Security disability and maintaining your benefits. And by popular demand, we have a program on orthostatic intolerance, which is part of dysautonomia or postural orthostatic tachycardia syndrome, POTS. Vasovagal syncope goes by a lot of different names, but Peter Rowe from Johns Hopkins University is coming on to talk with us about managing orthostatic intolerance, which from past programs is a topic of also great interest in the community, and some of the most disabling symptoms on an ongoing basis of CFS. And then I have a typo here on August, it's not actually even August 19th, it is September 16th. We have Tony Komaroff from Harvard Medical School coming on to talk about CFS and the viral connection, and he will include XMRV in that presentation, but won't limit it to XMRV. So there were some questions about enterovirus, HHV6, EBV, Q fever. I think Tony will cover a wide range of infectious agents that have at one time or another been linked to CFS. So look for that announcement as well, and we will follow up with an email so that you have a link to where to register for these programs. And just some URLs that you can find resources on our website and our mission statement. So I'll add my thanks to Suzanne, to Vincent and Cindy, and to our audience. It's still quite large despite the technical problems and disappointment of not being able to hear from Cindy on the Q&A. But we appreciate everybody's time this afternoon and taking part in this webinar series, and the great questions that came in both in advance and during the program. So for this episode of the CFIDS Association's webinar series, we'll close out and look for the recording and slides online. Thanks so much, everybody.
Thank you.
Thank you.